Good morning to all of you. Today, the another sub point in a unit number 2, that is the crossing over. In a crossing over, we'll see the recombination or crossing over, their definition, types, mechanism of crossing over, and significance. It is a process of incomplete linkage is known as crossing over. It is the exchange of chromatic segment between a homologous chromosomes. It results in the recombination of linked genes. It is the tendency for the genes to enter the gametes in a new combinations which are quite different from those of the parents. It is the mechanism by which a group of genes in a one chromosome sometimes change the place with the similar group of the gene on a homologous chromosome. We will see the definition, a mechanism of a recombination of genes due to interchange of chromosomal segment at the time of pairing, or it is the process that produce a new combinations of genes by interchanging corresponding segments between a non-sister chromatids of homologous chromosomes, or when a crossing over occurs in a body cells or somatic cells during the mitosis, it is called as somatic or mitotic crossing over. When it occurs in a sex cells or a germinal cells during the meiosis is known as germinal or meiotic crossing over. The crossing over occurs during the Pachetin stage of first prophase of a meiosis. Cytological proof for a recombination or a cytological evidence of crossing over. Carlston in 1931 gave the cytological proof for a recombination in Drosophila. He used two strains of Drosophila. Each had a special X chromosome so that they could be identified morphologically from each other and also from a normal X chromosome. To distinguish two X chromosomes of the female, he placed a portion of Y chromosome on one X chromosome. Other chromosome was broken into two pieces. The upper part of X chromosome had two genes. These are the mutant or sex-linked genes. In a Drosophila, he selected two strains, that is the carnation color of eye and bar shape of eye, that is the carnation is recessive to red color, while the bar is dominant to round, that is the plus B shape. Another X chromosome has attached a piece of Y chromosome. There were two normal genes, that is the plus or plus B. So this female fly was with a car B plus B genotype. This fly was crossed with a male fly having a car plus B character, that is the both recessive genes on X chromosome. The carnation color of eye and round shape of eye. In the above cross, the female fly produces four types of the eggs due to crossing over. Only the female progeny was studied. Out of these, 50% females were a non-crossing over and the remaining 50% females were a crossover. They are as, that is the first one, carnation bar, that is the car and capital B, one broken X and another normal X chromosome from the male. In a second one, a red round, which is the plus B, one X chromosome with attached Y chromosome and another normal X chromosome from a male. In a third one, a red bar, that is the plus capital B, one broken X chromosome with attached Y chromosome, other normal X chromosome, while the fourth one, carnation round, that is the car plus B, unbroken X without a Y chromosome and other normal X chromosome. Another four kinds of the male flies will be produced due to fertilization by Y carrying male gametes. Thus, of the four types of the first two belongs to non-crossing over types. Such a cytological observation suggests that the genetic crossing over takes place within an actual exchange of chromosome segments. In the non-crossing over, the X chromosome was as it is, that is either broken into two parts or entire with attached Y chromosome. In a crossover type, the maternal X chromosome is resulted from a crossing over, that is the red bar eyes, while the other X chromosome is normal, that is the carnation round. Thus, in this experiment, the genetic crossing over is proved with a cytological crossing over where the actual exchange of segment of homologous chromosome takes place. Thank you.